कैसे हैं आप सभी? आई होप यू आर आप लोग अच्छे ही होंगे। तो हम लोग डेयरी माइक्रोबायोलॉजी कर रहे थे। तो आज का हमारा टॉपिक है माइक्रोबियल एग्जामिनेशन ऑफ मिल्क। तो स्टार्ट करते हैं। इट कैन बी डन बाय द फॉलोइंग मेथड्स: स्टैंडर्ड प्लेट काउंट, फर्स्ट मेथड, और भी बहुत सारे मेथड्स हैं, हम लोग बात करते रहेंगे कवर। It is also known as variable count methods, which examines the variable bacteria present in the milk. It gives a rough and direct assessment of a variable number of bacteria and a clearly simple method to carry out. इसका procedure: take 1 ml of milk sample, then prepare serial dilution by transferring 1 ml of a sample from each tube, that is 10 to 100 times dilution, etc. Spread 0.5 ml of a milk sample occur to solidify agar medium. At last, incubate the prepared plates for 24 to 48 hours at 35 to 37 degrees Celsius and finally count the number of colonies. Coliform count. Next, examination. It is used to examine the presence of coliform bacteria, which can cause the fermentation of milk by the production of acid and gas. It becomes necessary to detect the presence of coliforms, as these make the milk unpotable for human consumption. Procedure: firstly, take McConkey fluid medium, then add milk of different concentration in fermentation tube. After treat, a same process like standard plate count. All dilutions are all the same, only media is different. Next test: methylene blue reduce test. It is rapid method to determine the microbial growth load. Using this test, we can identify the quantity of milk depending upon the color retaining property, the speed of reduction of methylene blue. Next is color is directly proportional to the volume of bacteria present in the milk sample. In simple words, an increase in the number of bacteria flora will reduce the color of methylene blue more rapidly due to the consumption of energy. Procedure: add a definite quality of methylene blue to 100 ml of milk. After that, hold the sample at 37 degrees Celsius until the color disappears. Next test is Reza burning test. It is very similar to the MBRT test. Procedure: firstly, add the Reza burning to the milk sample, then incubate the tubes for 10 minutes and observe the shades of color. Next test is phosphate test. It is used to check the pasteurization process to verify whether milk is pasteurized or not. Phospholipase is an enzyme that is usually present in the milk. This enzyme becomes inactivated if pasteurization is performed properly. Procedure: first, make 5 ml of milk in a sterile test tube, then add a few drops of sodium diphenyl phosphate. After that, incubate the tubes for 10 to 15 minutes. Enzyme cleaves phosphate groups from substrate and the lipophenol phenol. Now phenol reacts with another color changing compounds which is added, that is 26 discolor phenol bromide to give blue color. Next test is turbidity test. It checks the sterilization process of milk, whether the milk is boiled correctly or not to the temperature prescribed for sterilization. It sterilized milk all the columnar heat proteins get precipitated. Procedure: first take 5 ml of sterilized milk, then add a few drops of accumulation sulfate and boil it in a water bath for 5 minutes. Next, direct microflora count. It is a rapid method for the microbial examinations to determine the cell type and morphological procedure. Firstly, take 0.01 ml of raw milk in glass slides and air dry. Then add 1 drop of methylene blue. At least count the bacterial consumptions. A colony counter. It counts both variable and non-variable cells. And next video we'll start classification of various groups of microorganisms associated with daily industries. Thank you.